The correlation between the ability to taste PTC and Sinigrin hydrate.
A project by Julie Spencer and Brayden Cooley. So let me tell you a little bit about the PTC gene. So this gene is located in the TAS2R38 protein, and it's found on the back of your tongue in the cilia. So this gene has been linked to the ability to taste bitter chemicals or not. A person's genotype can be one of three genotypes. It can be a homozygous GG, which is an extreme taster, a heterozygous GC, which is a mild taster, or a homozygous CC, which is a non-taster. So if humans were put into a natural selection situation as animals are, you would want your genotype to be homozygous GG because you would be an extreme taster, you would be sensitive to bitter chemicals, and it would mean that you could sense, potentially poisonous chemicals in the environment, as opposed to someone with a CC genotype. So previous studies suggest that tasters and super tasters also differ from the non-tasters in just their taste preference and in patterns of food rejection and food acceptance. So we hypothesized that there would be a correlation between individuals who were homozygous GG, which are extreme tasters, and they would have sensitivity to the bitter glucosinolate chemicals such as Sinigrin.
So basically the purpose of this research was to study the effects of one's ability to taste a bitter compound called phenolthiocarbonide and the correlation it may or may not have with Sinigrin, which is a glucosinolate chemical that's found in raw Brussels sprouts and other plants in the brassica family.
Okay, so we hypothesized that there would be a correlation between individuals who were homozygous GG and the sensitivity to bitter glucosinolate chemicals such as Sinigrin.
So basically the question that we were asking ourselves during our experiment is Sinigrin tasting ability related to the ability to taste PTC in the PTC genotype? So we tested 76 samples, and first we genotyped them. In the PCR machine, we used 3 negative controls. We had 3 of those per run. And for the test samples, we only could do run due to money and time constraints. And again, we used 76 samples of those. And so for the phenotyping step, we had negative controls again. They were control paper. We had them taste it and rate it on a scale of 1 to 3. 1 being not bitter and 3 being extremely bitter. And we did the same for PTC test strips as well as Sinigrin strips.
Okay, so let's talk about our methods and procedures. As we said before, we obtained 76 subjects between the ages of 15 and 18. So we had the students rinse out their mouths to ensure a clean mouth and a clean palate. We blindfolded them so that they didn't know exactly what they were tasting. We distributed the control paper, and we had them put it on the back of their tongue, where those receptors are. And then we had them rate the taste on a bitterness scale from 1 to 3, and then we had them rinse their mouth again. We repeated the following bullet for the PTC strip and for the Sinigrin strips.
And so, on a continuation of our methods and procedures, this is how we extracted the DNA for the genotyping. So we had each subject use a cotton swab, or actually a sterile swab, in which they rubbed on the side of their cheek to take all the cells from their mouth and put on the swab, and then they swirled it in lysis solution, added stabilizing, we put it through the water bath, and then we set up the master mix for our genotyping, which is located on the bottom of the screen.
So a continuation of our methods and procedures. This is what we did for real-time PCR genotyping. So we loaded the 48 reaction wells of the PCR tubes with our master mix, and then with each subject's DNA. And we ran the reactions using the real-time PCR machine, which is a Step 1 Plus instrument, and we really enjoyed this step, as you can see from the pictures. And then we analyzed the results in Microsoft Excel. using tables and averaging to find the correlation or lack of correlation.
So this is a table of our results, and so the blue column are the sample's numbers. The column next to that call is what their genotyping was from the PCR machine that they used from their own DNA. Right next to that in the grayish column is the PTC phenotype, which is what they ranked the PTC test strips on a scale of 1 to 3, again 1 being not bitter and 3 being extremely bitter. And then on the column next to that in the white one is the Sinigrin phenotypes. And again, they rank that on a scale of 1 to 3.
I just wanted to make sure that we were locked
Julie,
in.
you're going to open up the door and shut off the sun.
Calm down,
It's closer to
Brayden.
the house.
Freaking A. So this slide is results continued, and it's just like a mini table of what you just saw in the last slide. It has the total numbers of what people rated for their Sinigrin taste, what each genotype rated on a scale of 1 to 3, and then we have also the same thing for the PTC taste, because we took the phenotypes also of each subject. So this next slide shows the correlation or lack of correlation between PTC genotype and Sinigrin phenotype. Since the number is less than 0.5, we can safely say that there is little or no correlation between the Sinigrin phenotype and between PTC genotype. Wah, 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 wah.
So, for the conclusion, it was originally hypothesized that the test subjects with genotype GG would rate a higher number on the bitterness scale than the genotype CC. And so this hypothesis was not true, we found out. And of the subjects genotype GG, 75% had the correct PTC phenotype, and only 33% had the correct Sinigrin phenotype. And so the rest of these bullets show the same data for the CG and CC, again with really low numbers for the correctness of the Sinigrin phenotype. So with this data, it can be concluded that there is very little correlation or almost no correlation between the ability to taste PTC and the ability to taste Sinigrin. Alright, on to the discussion. So this research brings up many questions. First, we determined that PTC genotype and Sinigrin phenotype would correlate, but our data proved that they do not, in fact, correlate. This could be due to the fact that the bitter taste receptors on the tongue that taste PTC only taste PTC and no other chemicals, so maybe further investigation can prove or try to find other taste receptors that can taste Sinigrin or other chemicals. Also, there were some limitations to our results, so there might be some confusion in the understanding between mild bitter and extreme bitter. Since everyone has their own opinions and it's hard to change those opinions, it can cause complications in the future. And to improve our research, we could possibly use a stronger concentration of Sinigrin hydrate, or we could use other chemicals that are responsible for bitterness in Brussels sprouts, such as one we found called the gluconostertin. And the only reason we never used this one is because it was very expensive and it was from Russia. I don't even know, and we don't even know if it was toxic or not, so we decided not to use it.
All in all, this experiment was an improvement from my experiment last year, which was to correlate PTC genotypes of test subjects with food preference. I found a correlation between PTC genotype and how much people liked Brussels sprouts and didn't like them. And so what I did was I took a chemical that is bitter and it's found in Brussels sprouts called Sinigrin, and that's why we tried to test just completely Sinigrin, the chemical itself, instead of adding texture with that and other chemicals found in Brussels sprouts. Some limitations we had with this experiment, as I said before, we were only able to test run subjects and genotype them only once due to money issues and also time issues. That was a big thing. So to make this project better, you could run the samples multiple times. It's time to acknowledge.
Woo!
So, I'd like to acknowledge my science teacher.
Henry Weeks. Ow!
for allowing us to use his classroom and his tools and also his students. And we'd like to acknowledge the students that posed as our test subjects.
Thanks for letting us bother you in your classes. Sorry.
And we'd also like to thank Life Technologies for providing the assays and helping with technical assistance.
Thank you very much.
Thank you, Life Technologies.
This is the end. We out!
Deuces!